welcome to our podcast on how functional genomics and proteomics help in sustainable agriculture and climate resilience. I am Yishan, and let me introduce my friend Sijie, Norman, and Yui. As we discussed, food demand rising fast. By 2050, we need more 50% food, while the worldwide will become 10 billion people. Meanwhile, the climate change will cause higher temperature, irregular rainfall, drought, flood, and salty soil, all of which will causing lower yields in plant. Plant respond with stress signal and hormone changes, but coping with several stress at once is still a mystery. Furthermore, the pests and weeds will become more resistant to antibiotic, and the overuse of antibiotic is also increasing this issue. The many staple food also lack of key nutrient, which will causing the hidden hunger. All of these challenges show us we need more modern genomics and proteomics tools to make our crops more stronger and healthier.
So now I have a question on how can actually genomics and also proteomics help to solve major global challenges, for example, like climate change and also in terms of food security?
Scientists can use the tools such as the genomic and proteomic, and genomic help to identify the key genes for those crops to make the genes more resistant to the high temperature and disease. Well, for the proteomic will show how this gene actually affect the protein. Then the breeder can use this scientific data for the cultivate crops, then to make more suitable plant to today's challenge, such as the disease resistant and climate change resistant, to meet the requirement of sustainable production, which is very helpful to ensure the future food security.
Alright, so building on what you said about genomic tools, I would like to understand how finding specific genes has already led to better crops. Can someone explain some successes in crop improvement?
Absolutely. Think of the plant DNA like a huge instruction books. The scientists will look through the books and spot the instruction, like which DNA or region are used to control certain traits, such as grain size, numbers, or how they survive well in drought. For example, in rice, the scientists found that there is a gene can make the grain size bigger and heavier. Thus, by choosing this good version of gene, the plant breeders can get more rice per plant. They also found another DNA region that help rice to handle the dry condition. Thus, the plant give better yields during the drought. Another region also help rice to cope with salty soil. Thus, they can keep the salt level balanced inside the plant. Finding this helpful gene means that the breeders don't guess. They just pick the desired gene and combine them into multiple traits, such as higher production with stress tolerance. Thus, the new crop can perform better under tough conditions.
That feels show the benefit, and I am curious, after this step that turn the genes discovery into the new variety, how do the researchers make use to find the marker information and other tools in the practice?
They start by measuring the traits of existing line and taking DNA sample. For non-helpful gene or DNA region, they use marker-assisted selection by checking which plant have desired DNA markers and selecting them. They also use genomic selection. Genomic selection is carried out by they collect many DNA markers across the genome and build a prediction model based on plant with known performance. This led them to pick the promising line earlier without waiting for a full growth of plant. After narrowing the candidate, the scientists will use gene editing tools such as CRISPR-Cas9 to adjust or modify the specific gene, for example, to make the grain size bigger. Throughout, the scientists will test and grow the plant under normal conditions or stress conditions to confirm the real improvement before moving on.
Wow, interesting. It's great to hear the methods. So what kinds of improvement have these combined tools produced in real tests or fields?
When used together, the gains are clear. The researcher found that the marker-assisted selection can causing the plant yields better in drought or salty soil when compared to original variants. Adding genomic selection can also speed up the finding of baseline, saving the server of time. While gene editing then fine-tune the traits. For example, changing a gene that controls grain size can increase the grain weight by about 20 to 30%. Field trials show these improved lines do better than the original lines under both stress and normal conditions, giving farmers more reliable harvests. Thus, by combining the marker tracking, genomic-wide prediction, and precise edit can create variety with higher yields and better stress tolerance. And all of these are the key for sustainable farming in changing climates.
Pests are actually insects that can damage or destroy many crops. Can someone give us an example of common crop pests?
Of course, the trip plasmid is the common pest on the cucumber and other crops, and then they will not only cause the plant necrosis but also spread the topo virus such as the groundnut bud necrosis virus, which seriously affect the crops yield. At present, most of the farmer now will use the chemical pesticide to prevent and control, but this will also pollute the soil and also will cause the pest to easily drug resistant. And now scientists are studying the fungi like Beauvania bassana as a more natural and environment friendly alternative. This fungi can directly affect the thrips, and then more importantly, genomic research has found that it contains a variety of pathogenic genes in its body, indicates that it has great application potential in the future biological prevention and control.
So how did scientists figure out how fungus fight the thrips?
It's quite straightforward. Researchers took adult melon thrips and exposed them to the leaf, which treated with the fungi. Then they also keep the control thrips on the untreated leaf. After that, they collect the thrip sample at different times, especially for 2 and 4 days after exposure begin. Then they extract the RNA and use the RNA sequencing to analyze which of the strip genes change the expression. And they sequence the RNA with Lumina High Sequence 2000 and analyze gene expression change, and key genes will then confirm by using the RT-qPCR. And then the results show the shift in immunity and metabolite, help us understand how the fungi affect the pest.
So the scientists used RNA sequencing. What did the data reveal that how the fungi attack the thrips?
The sample taken after 4 days of infection show a total of 1,141 genes will downregulate, while for 142 genes were upregulate. The data show the fungi significance inhibit several key processes in the thrips. Among them, the most important thing is the inhibition of lysosomal function, which is more important system responsible for the waste treatment and defense in the thrip cell. At the same time, it also inhibits the biosynthesis of insect hormone, which are crucial to the growth and development of thrips. In other words, Beauveria bassana JEF 350 play a role by blocking the key cell function in the thrips, and which prevent the pest from growing, defending, and maintaining a normal balance in the body. We have talked about how the genomic help to improve the crops, but for the farmer, it does help the farmer to earn more money or improve their life.
Yes. For example, cotton with a special gene can boost the productivity by 24%, while the profit can increase about 50%, which allow the farmer to invest more in their families and varieties. While in rice, the drought-tolerant varieties can give about 900 kilograms until 2,500 kilograms per hectare in the dry seasons, which means that it is extra about 15 to 30%. That means steadier harvest and income even at a very tough condition. And I'm curious that beyond the money, does this improved crop help in environment too, like reduce the use of chemical or water?
Definitely. Stress tolerance crops need less water and often less fertilizer, reduce the stress on the water source and reducing the runoff. Better pest resistance mean which use the few pesticide spray which can protect the beneficial insect and keep the soil and water clean. Overall, this crop help farmer and also support the healthier ecosystem.
So basically, biotic stress is definitely a major issue when it comes to in terms of the pest control in agriculture. But what about in terms of abiotic stress? Can it be just as serious for the crop?
Yeah, absolutely. Abiotic stress can be just as harmful as pest and disease. It includes drought, extreme temperature, salinity, and imbalanced nutrients that can negatively impact plant growth and productivity. In fact, Chang and his college in 2020 found that such stress can reduce average crop yield by up to 70%, which means the farmer are only getting around 30% of the plant is really capable of. And this is a massive gap when we are talking about feeding a growing global populations. So, since plant is literally rooted in plants, they evolve into highly complex system to sense and respond to stress by activating defense genes, producing protective protein, and even alter the visible threats. But here is the catch: responding to stress costs plant energy. So when a plant defends itself, it will grow less. And there is a tricky part in developing stress-tolerant crop, which is how to balance between survival with productivity. And that is where tools like proteomics come into play by giving the researchers the ability to study how specific protein behave under stress and finds way to fine-tune that balance.
Well, now we know proteomics works to resolve this issue conceptually, but is there any actual people working on this by observing how plants respond under stress conditions using this technique?
Yeah, definitely. For example, Maturity and her research team study how spring weeds respond to both normal and drought conditions. They measure plant stress like chlorophyll content, leaf temperature, and the water level in leaf to confirm they were stressed. Then they collected the leaf, extract the protein, and use liquid chromatography mass spectrometry to identify which protein have changed because of the stress. Finally, they compare the result to see which proteins might be helping the plant to cope with stress and survive.
So as we found out which protein are differentiate expressed under those two conditions, then we would like to know which protein are important in this context and work on them from better tolerance, right?
Yeah, exactly. Once we know which protein are affected by stress, as those involved in photosynthesis or protecting cells, then we can target them in breeding or genetic research. For example, Rubisco activates or glutathione as transferase help with drought survival. Then scientists can try to enhance their expression in crops. So proteomics is not just about understanding stress, it's also about finding practical target to develop stronger and more resilient crops.
So both biotic and abiotic contribute to serious negative effect to plants. But what can be used to guarantee the food quality and safety for consumer?
Yes, correct. So basically, both biotic and abiotic factor contribute negative effects to the agriculture. And in order to guarantee in terms of food quality and also safety is to using proteomic tools. So this is because basically proteomics tools can provide information on the nutritional profiles and also the protein content of the crop, as well as the molecular defense mechanism for the plants to overcome the stressor and to increase yield production. So in terms of yield production, currently there are many other ways, much more easier way to increase the yield. For example, conventional breeding and genetic engineering modification can increase the yield. However, they cannot actually identify precisely where is the functional proteins that express the phenotype, for example, higher protein content in the milk. So basically, proteomics in current stage in food quality and safety, it helps to trace targeted therapy by identifying the storage of protein, allergenic protein, and stress-related protein through proteomic analysis.
So how proteomic tools actually help to trace the targeted protein and how it contributes to food quality and safety?
So basically there are two methods for proteomic analysis, which are top-down and bottom-up. So the major difference between these two, for example, top-down analysis of the directly of one big protein. So on the other hand, the bottom-up and proteomic analysis, which involve from one big protein, you need to break down into much more smaller fragments first. So after that, we'll be involving for the purification of protein and also separation of the protein to analyze. And the most important thing is to compare using bioinformatic tools. So in the role of bioinformatic tools in current situation is to identify and compare the function of the proteins. For example, what allergenic protein have been detected, and you need to compare with other protein, which protein that actually produce much more lower allergenicity.
Since you mentioned combination of proteomic tools with the bioinformatic help to identify the allergy, currently, is there any successful experiment in food?
Yes, there are where the research is done by Halima and the research team. So basically, they discovered a few allergen-like protein using nano-LC techniques and the high-resolution mass spectrometry. So in the lentil seed, for example, like albumin-2 and endosynthetase. And from this, since they already know which protein, so they can compare which other lentil cultivars can produce much more lower allergenicity. So another research was done by El Salam, which they're using high-performance liquid chromatography called mass spectrometry and the MAUDI-TOF to actually assess the changes of the protein during breastfeeding of the milk, contamination, preservatives, and also the allergens.
Now that I have heard how proteomic help with plant trace and food safety, and for the genomic improve the crops and pest control, I see they are powerful tools, but how do they connect to sustainable agriculture?
Well, in my opinions, by using tools like next generation sequencing, molecular marker, or NAI, scientists can actually understand a plant DNA and identify traits like drought tolerance, disease resistant, or high yield. This means we can actually breed crops that can grow well even in tough conditions by using less chemical, less natural resources, and less water. So that's it. Besides improving crops, genomics also protecting the environment, reducing waste, and ensuring we can feed our future generation in a more sustainable way.
So just now Norman had mentioned in genomic terms, and how about in terms of proteomics that help in sustainable agriculture?
So basically, proteomic in sustainable agriculture can be achieved by using the proteomic tools, for example, like mass spectrophotometry, gel electrophoresis, and most importantly, with the combination of the biometric informatic tools to actually identify and predict the allergens, and also the early identification of the protein that are responsible for stress and also disease in agriculture. So from this, since we already know which protein that causes to the plant production, hence we can either, for example, delete them or maybe replace them with other beneficial protein that can produce better result, for example, better production. So by using these proteomic tools, not only help in better yield production, and most importantly, it can help to reduce waste and less environmental damage to ensure sustainable agriculture.
Okay, I think that's all in our podcast, and thank you for watching our podcast.
Remember to like and share.
Comment.
Okay, bye-bye.
Bye bye. Higher temperature, irregular rainfall, and drought. Sorry.
Researcher took the adult mon.
Alright, so based on 不是 then